And now we have Kathy Zackowski. Dr. Zackowski is going to talk to us about the use of transcranial magnetic stimulation in transverse myelitis research. Thank you.
Hi, everybody. I'm Kathy. First of all, it's not going to be boring, even though I know the title is not very stupid. I almost took that title off because I'm like, that's a mouthful, and it's right before lunch. So, so bear with me. The idea is really to give you a flavor for what this kind of technique can provide to the TM community, and I'm happy to take questions. And I wanted to sort of bring up, so I'm on faculty here, and I'm an occupational therapist, and my overall goal is really to try to bridge this gap between what we know about basic science to how we can actually change what we do to treat individuals that have conditions like TM. So not only with medications, but also with rehabilitation, because I think understanding this link is really critical. And that's the focus of my talk. That's why I think this technique is very interesting for TM. So I'll go on from there. So I'll talk to you about, I'll define transcranial magnetic stimulation. I'll call it TMS from here on out. I'll tell you about some applications, both for diagnosing and for therapy. And then I'll talk about some studies that have been done in people who have TM and spinal cord damage. So as Dr. Becker said earlier, in the last few decades, our view of what the central nervous system can do has really changed. So we formerly thought it was kind of this rigid structure, not very changeable. What you have as an adult is what you get. But now, thankfully, we know that it's very changeable, and we call it plastic. And by that, we just mean it can be changed by activity, it can be changed by medication, by stimulating the system is how you can change it. We know this change in how we understand the CNS to function has partly occurred because of all the technological advances. So we use high technology like MRI and PET scans and transcranial magnetic stimulation to better understand how the output's going to be changed. So we need a window into the central nervous system, and these high technology devices can give us that window. So TMS is a tool that can be used to study broadly the connectivity of neuronal pathways. So how we know that in TM that, as Dr. Becker explained, you lose some myelin, and so the message from your brain to your muscles is sometimes slowed down, sometimes lost completely. And so we can use TMS to try to quantify that change. That's what I mean by connectivity of the neuronal pathways. We can also use TMS to evaluate the plasticity, so how changeable are the neurons in each person's body. So there's differences from person to person. We can also understand or try to use TMS to better understand the behavioral consequences. So how does using TMS change the way you move? That's really what we want to get to, right? It's all great to understand the biology, but if it doesn't make an impact to how we run our life, then that's not as much interest. So we want to bridge this gap between the biology and what we do in our daily life. So the use of TMS it's one method of looking at the electrophysiology of our body, and this could indicate the underlying system or pathways that are affected and provide the scientists with insight into what is the mechanism of action. And that's important because that's how we devise medications and test them, and that's how we devise rehabilitation techniques. So I'm showing here a picture of, this is a figure-8 coil, and this is the box that essentially provides the energy to this coil. So this coil will be put on a person's head, and it induces a change in the magnetic field. So this box essentially puts out a electrical pulse. You put it on your head where you want it. So there's a part of our brain that has all of the motor signals that go down the spinal cord. So you put it there. You elicit this magnetic field, which causes the neurons in your brain to fire. And these can be excitatory or inhibitory. So I'm showing here an example. So this is the coil that we put on the back of a person's brain. As you turn it on, there's a spread of that magnetic field through your brain. And we know that as you go further out from where that coil is, it goes, the TMS field, which is demonstrated here, gets smaller. So this is the distance that the signal will travel once you put this stimulator on, and this is how much of an electrical field you'll emit. So you can imagine that it's stronger here than it is there. That's basically all this is telling you. And that it's important to recognize that this is a great tool because now we can elicit changes in the neurons that go all the way up to here, right? But we're not just changing the neurons that are excitatory or causing more movement. We're also exciting the neurons that are damping things down. So it's the total composite of that activity. Now this I got from, so Dr. Selnick shares the lab with me, and he does a lot of TMS, mostly with individuals who have stroke. So he gave me this slide, and I like it because it gives you the idea of what TMS is doing. So let me explain it before I run it. So here's the coil. You put it on your head, you're going to stimulate the neurons, then that signal is going to go through your brain. This is the spinal cord, small as it is here, and then it goes out, and you can actually measure a change in a muscle. So the important thing is you stimulate up here, but what you're really measuring is the output. Again, that's what's important, right? How well can we use our limbs? And so what we measure out here is called a motor evoked potential, or an MEP. And I'll keep coming back to that, because that's our measure that this stimulation did something. How well did it get down to this muscle of interest down here? Now, this was similar to peripheral nerve studies. The amount of time it takes to get from stimulating here to down out here to the muscle is important. So you can stimulate up here, or where the motor cortex is. You can stimulate down lower in the brainstem, and you can even stimulate at the top part of the spinal cord. And that's important because it gives us an idea of how long did that signal take to get from wherever we stimulated out to the muscle. That tells us something about how healthy the tissue is, how healthy that pathway is. Now, TMS is used as a diagnostic tool, and what we do is we measure, and we have all of these different variables that give us little pieces of information about how healthy the spinal cord and brain are. We also use it to evaluate therapies. So how plastic is the brain? Can we actually change behavior? So I'm going to define some of these for you. I'm doing it in kind of a very general way, so if you have questions about specifics, I'm happy to take those, but I wanted to just get the main point across to you today. So motor threshold, that's something we measure a lot. All that really means is what is the least amount of stimulation I can give a person and get a motor evoked potential. And we know that medications can actually change your motor threshold. So that's important to know because with TM there are medications that are used as well. So this could change how effective TMS is. There's also something called a recruitment curve. So that's important to me because I want to know when I stimulate with TMS, here I'm doing it at 100% and then increasing up to 150, I want to know what that MEP amplitude does. And if I build a curve like this, I can get more information about what the entire network excitability is like. So that's another measure that I use a lot. There's also something called intracortical excitability. So you give a test pulse. So I didn't mention this, and I will. When you use this coil, there are single pulses. So there's just a single pulse for about a millisecond that is provided, and that's what gives you this single MEP wave. So you give a test pulse. Now, it turns out that if you give 2 in a row kind of quickly, that is considered inhibitory. So you can see that the signal is smaller, right? If you give them 2 pulses a little further apart, now it's excitatory. So I can modify what kind of stim I give and get different information. And this is important because, so this is inhibitory here. It's called short intracortical inhibition. This is facilitatory, which is making it more excitable. That's called short intracortical facilitation. And that's important because that relates to neurotransmitters in the brain and spinal cord. GABA is the main inhibitory neurotransmitter in our brain, and glutamate is the main excitatory. And we know that changing this interval changes how the MEP responds. Okay, so that's sort of the basics very quickly about TMS. So now, why is this important to TM? It turns out there's not a lot of studies that have done this in TM. So what I've done is there's one study of TM and there's 2 that I took out that have spinal cord injury that I think are similar enough. I didn't mention this at the beginning. I changed my slides slightly, so it's not that you got the wrong version. I didn't bring this one till just now, but I didn't make a lot of changes, but this should make it a little clearer to you, I'm hoping. So there's one study that was done that looked at an individual who had a lesion in their spinal cord. You can see it here. This is as though you cut my spinal cord like this and kind of laid it out. That's what you're looking at. This little part here, this little white part shouldn't be there. It should look all gray. This is a sagittal view like Dr. Becker showed, where this is your spinal cord. This part here and this little white area is not supposed to be there. That indicates damage. So in this study, what they did is they said, okay, this person has a lesion in a very specific area of the spinal cord that just affects the fibers that carry sensory information. So you would predict that they should be okay motor-wise. They should be able to walk because it's just the sensory fibers that are damaged. But what they found is that even in an isolated lesion that's just affecting the sensory fibers, there are changes, long-term changes that are happening up here in the brain that have the motor fibers. So that's important from a rehab point of view because that suggests maybe we should emphasize sensory training, because if we know lesions that are just affecting sensation, if we can stimulate those sensory fibers, maybe the motor cortex will improve as well. Now, a separate study looked at, again, a child who has transverse myelitis and followed the recovery and followed the child came in, can't walk, has serious sensory loss as well. And they did TMS and looked at 5 different muscles. And I apologize, they're cut off. I didn't realize that until I put it up just now. So 5 different leg muscles. And what we're showing here is the left and right side MEP. So this is the motor evoked potential for these different muscles. This is at baseline, this is at week 6, and this is at week 12. So the overriding message, right, is that, look, these signals now get bigger as you go further from the time of damage, right? So this is great because the MEPs are now becoming more active, which suggests the muscles are more active. And it turns out that in this child, over 12 weeks, their walking improved substantially. This is important because the motor recovery paralleled the MEP improvements. So maybe TMS can offer a way to monitor this and monitor not just the function, but what's changing really in the brain and spinal cord. Now, my last study that I'm going to talk to you about looked at, this is individuals who have spinal cord damage, and what they did was they used TMS to monitor change after people have been through rehab. So one group had conventional physical and occupational therapy. The other group had special rehab where they used robotic arms. This is an arm study for right now. So they used a robotic arm to facilitate more and more movement. And they followed them over 6 weeks, I believe. And what I'm going to show you here is this is time on the x-axis. So baseline measure, everyone's at 0. Everyone was normalized, okay? Week 2, week 4, and week 6. This is how big that MEP, how much it changed. So not the size, but the improvement of that MEP size. And what you see is that 2 lines go up, right, consistently. This is the biceps, and this is the wrist extensors. And they improved specifically on this special kind of therapy, this robotic therapy, which I'm being kind of vague about because I'm not sure that's the message. The message is that there was improvement with rehabilitation and in specific muscles. Now, recognize that there's not improvement in all muscles, so that's a curious question. Why are specific muscles being more improved in this study and others are not? But the take-home message I'd like you to take is that, again, motor recovery paralleled what we see in the MEP amplitude. But with the limitation that this is a group study, so at least it's not a single person again, but it's a group of 13 individuals. So that's a small sample. And what we really need to move rehabilitation forward is we need rehabilitation research that looks at larger groups of people. And that's important. And it's important if you want to be involved in research studies as a participant that you understand the point of what they're doing and that you agree with the point of the research project. So what I've shown you so far is that TMS can be used to understand brain processes. It can also be used to induce plastic changes, which can modulate behavior or motor function. TMS can also be used to help understand the basic physiologic mechanisms that are associated with motor behavior. And this has allowed us to develop novel strategies to enhance recovery processes. Much of this research that I don't show you here to sort of support this comment is done in individuals who have a stroke. So that just means they have a lesion in their brain, more likely than in their spinal cord. But there's no reason to think this couldn't be translated to TM as well. So how can we use this technique to provide additional information about TM? So here's our plug. So maybe we can evaluate the effectiveness of a known pharmacologic therapy. And that's what we're doing at Hopkins. So I'm going to tell you about our new study with the idea that I'm informing you of what's going on. And if you're interested, you know who to talk to, because all of the people involved are here today. So the study is titled The Efficacy of Sustained Release Oral Dalfampridine in Transverse Myelitis. That's a mouthful. I'll explain what all that means. It's happening here. The investigators are myself, as well as Dr. Becker, Dr. Pardo, and Dr. Levy. So let's backtrack a little. So I don't know how many of you know about fampridine or 4-aminopyridine. It's a medication that's been out for a long time. What it's known to do is it amplifies how conductive those neurons that have lost myelin are. And so that can provide a nice therapeutic benefit. But what has been found is that the therapeutic benefit of using this medication, this specific one, is limited because you can take too much, and that increases your likelihood of getting a seizure. So who wants to take that, right? So it's got limited usefulness. So what was done is they developed a modified version of this drug. So what the drug does, if this is an axon, like Dr. Becker showed, these are little sections of myelin. In NTM, you've lost a couple of sections in here, right? And so what happens when you lose that myelin is that there are little channels that have potassium going in and out of the cell, and it opens all of those channels up, and that makes it hard for the signal to jump from one myelin node to the next, okay? So what happens with this drug, this new oral dalfampridine, which is a modified version of this drug, is it plugs some of those channels up. It blocks potassium channels, and that helps nerves to recover. So what do we know about dalfampridine's effects? So there was a large study done on individuals who have multiple sclerosis. So a different diagnosis, but they're still, people with MS are also losing myelin, right? So what they did is they brought individuals with MS in, gave them a placebo for 2 weeks, and then separated them. Gave some of these individuals fampridine, and then gave other individuals a placebo, and didn't tell them which was which. Kept them on this medication and measured their walking speed for 14 weeks, and then took everybody off and followed them for another 4 weeks. So no medication. Let me show you what they found. So I'm going to point out, so the x-axis is time, so 14 weeks is from visit 3 to visit 6, and then the follow-up time's here. And this is the change from baseline, so how much walking speed improved. The open blue, I'm going to go with the most boring part first. The placebo group didn't change throughout the study. Not a surprise, right? The light blue group, these people did get fampridine, but they didn't respond as well for some reason. So their walking speed improved slightly at visit 3, but then it kind of plateaued out. But interestingly, when they were taken off the drug, now they went down again, back to where their baseline was. But a group of individuals, only about 25% of individuals, but a group of them were considered fampridine responders. So they responded a lot, and their walking speed improved substantially and was maintained over 14 weeks. And when they were taken off the drug, it plummeted, suggesting the drug is really causing this change, right? So what we thought, what this shows, let me back up a little, is that dalfampridine, this sustained release potassium channel blocker, so now you have less risk of seizures, right? Because now it's a time release, so you can't get too much, has been shown to be effective in improving walking in individuals with MS. Dalfampridine also has the potential to improve gait and neurologic function in patients with TM because there's a similar pathology with MS. Oh, sorry, not MD. So what do we hypothesize? We hypothesize that this drug, dalfampridine, will improve walking and neurologic function in patients with TM, and that the use of transcranial magnetic stimulation, see the full circle coming around, will improve our understanding of what the characteristics are for who are these responders. Because why is it only 25%? What's different about them? It'd be really nice to know that so we could just target it to those people and then be working on different medications for another group, right? So here's our general procedure. So we're going to have a similar 2-week placebo run-in, and then we're going to divide people and follow them for 8 weeks. Some will get dalfampridine, some will get a placebo, and they won't know, and I don't know either. And then they'll be crossed over, so that the people who got dalfampridine will now be put on a placebo, and the people who are on the placebo will now get dalfampridine. This keeps everybody happy, right? Because then everyone gets a chance to try this. But we still get to see if it works or not, and we'll also follow them after we take them off the medication. So I wanted to put up sort of the basic inclusion criteria. There's kind of a whole process to being part of a research study, but at least to give you an idea, you basically have to have a diagnosis of TM. You have to have some problems walking, and we'll define that by how quickly you walk your 25-foot walk. And you have to be between the ages of 18 and 70. And I'm happy to answer questions now and throughout the day. And then I put Maureen Mealy's email down here, and you can contact her if you have questions as well. And Dr. Levy and Dr. Becker are here as well, so we all are happy to answer questions. And I don't want you to feel like I'm telling you this because I'm saying everyone should be part of this, but I wanted to really bring around this whole idea that the more we understand the mechanism, the better we're going to be able to treat TM. And so these kinds of studies are what we need to improve the outcome. So that's it, and thank you for your support.
Couple questions.
Or do you want to eat? Is that the everyone's?
But if you want to eat, there's lots of opportunity for questions later, as always.
Why the age? I would have made one of these before. Why the age limitation over 70?
So if there was someone who was over 70 that really wanted to participate, we might we can always go back to our IRB and change that. But we thought we wanted to maintain sort of a cohesive group that is an overall general health. I think it was sort of a general requirement, but I don't think it's set in stone. Is that? Yeah. Yeah.
As you see the improvement in these individuals, what happens with their pain?
Very good question. So the question is, as you see improvement with people, for example, with dalfampridine, you're saying, and what happens with their pain? So it's a really important question because pain in the MS study, you can correct me if I'm wrong, I don't think they monitored it or saw a big change if they did monitor it because it wasn't a main finding. So that would be something we would, in TM, I know pain can be an issue, so we're going to monitor that and see if there is change and keep track of that. That's kind of the best I can offer. I know it's kind of a lame response, but that's what we have.
So you said potassium, potassium disrupts connectivity. So
Right.
You can't just increase potassium, or would that seizure and
So we lose potassium out of the axon because of the loss of myelin. So just eating more potassium won't change that. It really is the fact that these potassium channels are losing potassium too quickly, and that disrupts the health of the axon overall. Yes. Oh.
How about for people that can't walk?
Good question. So I think at this point we had to limit it to the time 25-foot walk because we wanted to really mimic what's been done in the past to really validate that we're using the same procedure where they found a response in MS patients. But I will say that there's no reason why it has to be walking. I can tell you from the MS patients I've seen on this medication, it changes their overall ability to move. It's a systemic drug, so it's not really just going to target those muscles you need only for walking.
Okay, we're gonna stop there, and then we do have a break now, and this is where you get food. But again, we're actually going to be addressing this trial again later. And then one of the study coordinator, Hua, can you kind of raise your hand? He's going to be set up actually right outside where you registered and is going to have pamphlets regarding this trial. See this pamphlet, and also my card with my contact information. So if you're interested, you can just email me with all your questions if any of them don't get addressed. But we're going to have
Let me tell you too, Hua is very quiet. It's very he's very good at what he does.
10 minutes, and so you can grab food. And also we're going to have some people bringing food to you. So if you don't feel like you need to get up, you don't need to.